Welcome to The Explainer. Today, we're going to decode a brilliant, life-saving biological strategy by zooming deep into your cells. I want you to view them not as static blobs, but as bustling, high-tech molecular factories. Think about the sheer scale of it for a second. Your cells are churning out thousands of proteins literally every single second, operating around the clock like a perfectly synchronized assembly line. But, biology is messy, and the universe loves chaos. Things don't always go according to plan. So here's the kicker. What happens when that microscopic factory faces a catastrophic meltdown? Imagine it suddenly runs out of raw materials, its intricate assembly lines physically break down, or even worse, hostile viruses actively invade the workspace and hijack the machinery. What does it do to survive? It can't exactly pick up a phone and call for outside tech support, right? It is entirely on its own. It has to rely on an incredibly sophisticated, built-in emergency protocol to prevent a total collapse. Here is our blueprint for today's factory tour. We'll cover 1. The cell factory in crisis. 2. 4 sentinels and detection. 3. Master switch EIF2 alpha. 4. The paradox of the ISR and ATF4. 5. Activating cellular survival programs. And finally, 6. The crossroads of survival and death. Let's get right into it. Starting off with section 1. The cell factory in crisis and those existential threats. Imagine trying to keep a massive cellular factory running at top speed during a severe crisis, like starvation or a huge pileup of misfolded proteins. If the cell just stubbornly pushes the assembly line forward while the machines are broken, it's going to waste precious energy, ATP, and create a massive amount of toxic molecular junk. That is a fast track to total, irreversible ruin. So, to avoid this, the cell kicks off a brilliant protocol called the integrated stress response, or ISR. This strategy is essentially a calculated decision to immediately hit the brakes. It halts regular production to conserve valuable resources and pivots entirely to damage control. It's a calculated pause designed to buy the factory just enough time to fix the underlying problem. Move into section 2, 4 sentinels and detection, or how the factory alarms actually work. Because you gotta wonder, how does the factory even know it's in trouble in the first place? Well, a facility of this unbelievable complexity needs a top-tier security system. It relies on 4 specialized alarm sensors, known biologically as kinases. Think of these as our dedicated, highly trained security guards. First up, PERC watches the main manufacturing floor, the ER for protein pileups. GCN2 is a totally different guard. It detects when the factory is starving for raw amino acids. PKR is your antiviral alarm, tripping the moment it spots foreign viral RNA sneaking in. And HRI, it monitors the heavy power plants, the mitochondria, looking for iron shortages or functional stress. So you've got 4 completely different existential threats monitored by 4 distinct alarms. Which brings us to section 3, the master switch, EIF2 alpha, and the ultimate bottleneck. Now, here is where biology gets incredibly efficient. Even though these 4 sentinels monitor completely different threats, from hostile viruses to basic starvation, they don't trigger 4 different chaotic responses that might conflict with each other. Instead, all these alarms converge on a single vital bottleneck. No matter which sentinel rings the alarm, the activated kinase reaches out and alters one specific protein, EIF2 alpha. It slaps a phosphate group onto a very specific spot called the serine 51 site. Think of this action as physically flipping the master breaker switch for the whole building. This single molecular tweak synthesizes all those diverse, unrelated stresses into one unified, undeniable chemical signal. So what happens when you flip that master switch? Well, the altered EIF2 alpha actually changes shape and acts like a molecular trap for another crucial machine called EIF2b. By trapping EIF2b, the cell stops the formation of something called the ternary complex. Now, if you think of the cell as an assembly line, the ternary complex is literally the starter engine. Without it, you absolutely cannot start building new proteins. By trapping the key to the starter engine, this master switch effectively kills power to the main production lines, stopping the bleeding and conserving massive amounts of energy. Section 4. The paradox of the ISR and our emergency responder, ATF4. And here we encounter a truly wild biological mystery, famously known as the ISR paradox. Just think about it logically for a second. If the cell just purposely shut down all of its assembly lines to save power, how on earth does it manufacture the specialized tools and machinery it needs to actually fix the broken factory? It's a real catch-22.
On one side of this dynamic, we see a massive intentional drop in global protein production. The factory goes completely dark. But on the flip side, the production of one specific hero protein, ATF4, dramatically spikes. It completely defies the general shutdown. The cell somehow selectively prioritizes creating this exact emergency responder while actively suppressing literally everything else.
How does it pull off this magic trick? The cell uses elements called UORFs, which are essentially clever little molecular traps placed right before the ATF4 blueprint. Under normal, healthy conditions, the fast-moving cellular machinery is racing along, clumsily falls into these traps, and fails to make ATF4, which is exactly what you want when there isn't an emergency, right? But during a crisis, when resources are heavily restricted, the machinery moves much more slowly and carefully. Because it's taking its time scanning the blueprint, it cleverly skips right past these traps and successfully initiates the synthesis of the ATF4 protein. It is an absolutely brilliant evolutionary fail-safe mechanism.
Section 5. Activating Cellular Survival Programs, or ATF4's Critical Mission. With ATF4 now successfully synthesized and deployed onto the factory floor, this emergency responder doesn't just wander around aimlessly. It heads straight to the central control room, the nucleus of the cell, to activate a massive, sweeping survival program. ATF4 becomes a powerful command hub. First, it rebuilds amino acid supplies. Then it deploys strong antioxidant shields to protect against damage, partnering with a crucial factor called NRF2. It secures energy stability by communicating directly with the mitochondria through a highly specific pathway known as the OMA1 DILI1 HRI axis. And, my personal favorite, it triggers autophagy, literally forcing the cell to eat and recycle its own broken machines and toxic junk. It is a complete, top-to-bottom operational overhaul.
Now, if ATF4's rescue mission is successful and the damage is repaired, the cell knows it's time to get back to business. It deploys a factor called GAD34, which effectively acts as the all-clear signal. This factor guides the PP1 enzyme, which you can think of as the factory's ultimate reset button, to reach in and delicately remove that initial phosphate group from our master switch, EIF2-alpha. The alarm is officially turned off, the factory lights come back on, and normal, healthy protein production safely restarts.
Finally, Section 6. The Crossroads of Survival and Death and the Ultimate Choice.
But, biology is pragmatic. It's not sentimental. We have to face the dark reality of a factory that just cannot be saved. What happens if the viral load is just too high, or the structural damage is simply too severe to fix? If the stress is chronic or overwhelming, the ISR makes a dark but necessary calculation. Our hero, ATF4, switches its entire strategy. It induces a specific factor called CHOP, which permanently triggers apoptosis, programmed cell death. The cell opts to hit the self-destruct button on its own factory. It sacrifices itself to prevent the spread of toxic, misfolded proteins or replicating viruses to the surrounding healthy tissue, ultimately protecting the organism as a whole. Ultimately, this whole journey into the integrated stress response reveals a profound biological wisdom encoded right into our very cells. When salvageable, mobilize all resources to repair. When impossible, gracefully shut down. Understanding this exact cellular pathway is actually helping scientists unlock completely new treatments for devastating conditions like cancer and neurodegenerative diseases today. So I leave you with this. Does knowing that your cells are constantly making these hidden, split-second life and death calculations inside you change how you view the incredible resilience of your own biology?